Vamos a dar un par de minutos en lo que vienen los demás. Muy buenos días a todos. Good morning. Welcome to this magnificent venue, which today shines even brighter, filled with the remarkable talent of our country across multiple health-related disciplines, students, professionals, and researchers. It is a true honor to host you here in a space where science takes center stage. I would also like to warmly acknowledge those joining us through the different channels and social media platforms of the Academy. Welcome, everyone. I wish to extend my deepest gratitude to the National Academy of Medicine of Mexico, in particular to its president, Dr. Raúl Carrillo Esper, and all the Academy management team, in particular Elizabeth. Gracias, Elizabeth. Gracias, chicos, por este maravilloso trabajo, for their invaluable support in making this symposium possible. I also extend a heartfelt welcome to the academics who are with us today, both in person and virtually. I'm especially grateful to Professor Adam Cunningham and Professor Burkhard Ludovic for accepting our invitation to share us some of their outstanding research journeys. Both have profoundly shaped the scientific history of my laboratory and, indeed, contributed to strengthening immunological research in Mexico. Over more than 2 decades of collaboration, young Mexican researchers have had the opportunity to train first in various laboratories here in Mexico and then continue their development in theirs. These collaborations have resulted not only in a significant number of publications in some of the world's most prestigious journals, such as Science, Nature Communications, Science Immunology, PNAS, among many others, but also in the creation of knowledge that endures through the researchers trained, opening new avenues of exploration in the vast field of immunology. For us, it is an honor to welcome them, and for me personally, it is a privilege to count them not only as collaborators, but also as friends. Throughout this symposium, we will hear just a few examples among many of the rich legacy that has emerged from these efforts. I invite you all to make the most of this forum of exchange, to share your ideas, reflections, and proposals, so that together we might continue placing knowledge at the service of society. Once again, thank you for being here, and it's my great pleasure to extend to you the warmest of welcomes. May this symposium be as successful for all. Es cuanto. So now, if you can introduce.
Yes. Hello, yeah, can you hear me? So it's a pleasure and an honor for me to be here and introduce you the talk of Dr. Adam Cunningham. So I had the pleasure to work during my PhD with Dr. Cunningham around 9 months and learn a lot about salmonella biology and infection. And it was actually one of the milestones of my career, so I'm really happy to meeting him here again in Mexico after so many years. So as a brief introduction, Dr. Adam Cunningham is a professor of functional immunity at the University of Birmingham, a director of Back-to-Back, a global network accelerating bacterial vaccine development. His research defines how adaptive immune responses are initiated, regulated, and sustained following infection or vaccination, with an emphasis on antibody responses to bacterial pathogens, especially in Salmonella. He was trained in pathobiology and immunology at the University of Southampton, and he established his independent group in Birmingham in 2005 and was appointed professor in 2011. His work informs the rational design and evaluation of next-generation vaccines to address antimicrobial resistance, and he will talk to us today about how the inflammatory response is induced to salmonella-derived thrombosis. So welcome, Adam, and it's a pleasure to have you here.
Thank you very much, and thank you very much to Constantina and Christina for that very kind words. And it's absolutely wonderful to be here in this really, really beautiful environment and to share some of our work with you. And before we do, I'm just going to plug the network that was mentioned about Back-to-Back, which is a network which promotes the development of bacterial vaccines. And although I'm talking about thrombosis here, it's really important to remember that if we have decent working vaccines, what I'm going to talk about next isn't a problem. Okay, so this slide is just to get my thanks in early. These are a group of people who've worked on this project and collaborated on this project over many years now, and I'm exceptionally grateful to all their efforts. I'd just like to point out Marisol and Noni, who both came from the excellent research pipeline, which is within Mexico. Okay. So bacterial infections remain a leading cause of death. One study published a couple of years ago now showed that estimated that about nearly 8 million deaths globally were caused by just 33 bacterial pathogens. This equated to about 1 in 7 of all deaths globally. And of course, our ability to do anything about this is becoming more and more restricted, both in terms of the treatments that we have available, but also where we can use those treatments globally, access and so on and so forth. And when we think about, why should we die from a bacterial infection? Why should we suffer? Why should we experience any symptoms? Sometimes it's really to understand it. If we take the causative agent of tetanus here, it produces an exotoxin, and that neurotoxin has a specific known target. You take away the toxin, you take away the problem in the main. For other organisms, like the gram-negative organism Salmonella typhimurium, it doesn't have an exotoxin. So it doesn't have that simple pathway to really drive its effects on us. It does have endotoxin, most often referred to as lipopolysaccharide. And if you look where you find LPS, it's in the gram-negative cell wall. And if we take a step back and ask what evil function that LPS performs, what does it do to us that so cruelly causes such harm to us, then all it does is make the cell wall stable. And so it's actually the way that we see LPS, how we respond to LPS, that dictates the harm that it causes to us, and not an intrinsic toxin activity of that molecule at all. And that's really important, because when we look at infection, we can think about things in terms of outcomes, always killing the organism, but what does it do to us? So we've got 3 scenarios here. Here's our bug, and here it's in the environment of you or me, and each time. If we have a vaccine, then the vaccine acts like a sniper, and it takes out the pathogen, but it leaves the rest of us intact. We're not harmed by this, and that's why we don't know when a vaccine is working in the main, because it's working silently in the background. Under normal circumstances, when we respond to an infection, we either bring out innate or adaptive immune responses, and these kill the bug, but also do a limited amount of harm to you as well. And finally, we can go nuclear on this. Now, the key point about all these three scenarios is that you're taking out the pathogen in each one of them. The immune system is fulfilling its role of killing the target pathogen. But the harm it does to you, these processes do to you on the way, they vary quite significantly. And so cross-cutting all of this is, of course, inflammation. And when we think about this, we often think about it in the sense of immune cells acting, or we think these are good. And then we also think about mediators, and these are cytokines. And here are a couple of ones I'll be talking about: interferon gamma and TNF. And then we also have other molecules like IgG. But actually, these don't work in isolation in the main. They work in a functional, organized manner. And this is incredibly important because the whole immune system is set up to be organized. And so we can see this here, whether we're thinking about it at a gross level in terms of, as we have 700 or so lymph nodes distributed throughout the body to prevent antigen dissemination, as you can see here. Or you can have other presentations of this, like bacteria being brought together in the form of an immune complex, whether the bacteria are intracellular, either in or outside of a vacuole. Or if we now start going up in scale a little bit more, we've got granulomas, or an area that I'll be mainly talking about here is the concept is if we encounter pathogens in the vasculature and that we try and trap these within thrombus. So all of these are not necessarily about instantly killing the organism, but they are about mounting controlled immune responses in selected regions to prevent bacterial dissemination or control bacterial dissemination. And if we think about it, even if we just take a very simplified view of this, inside our gut, we have roughly a kilo or so of bacteria. But we don't necessarily care about mounting fulminant responses against those organisms because we have they are not affecting us systemically in that sort of simplified manner. Okay, so what I'm going to talk about today is the concept that all these encounters lead to impacts on the cardiovascular system. And probably if there is a couple of messages to really take home from this is this is a really important statement from Steve, who I showed you the picture earlier, is that nothing kills you faster than a platelet. Literally, you can be dead inside a few seconds from the consequences of these becoming activated. And that's important. So if you start joining a few dots together of what I introduced in the last couple of slides in this, is that we might think we don't have to kill every organism that we ever encounter. Our gut bacteria show us that that's the case. But we do need to control where they are. And this both buys you time to mount adequate immune responses, but it also means is that other activities like the cardiovascular system can go on and do its job, which is to keep you. So our immune systems in this context are acting both as a police officer to police what's going on inside normal physiological activities like the cardiovascular system, but also as the army to protect you from external threats. Okay. So we know that pathogens and their products interact with the cardiovascular system. And one of the concepts of this is around this immunothrombosis, which is generally seen as being positive. Pathogens interact with various components of the immune system, resulting in platelet activation. You get pathogen trapping in that sort of classical model I showed you in a simplified cartoon earlier. But then you can also have thromboinflammation. And here, what you're really looking at is more of a dysregulated inflammatory response, where you get pathological clot formation and essentially cascading of inflammatory pathways. And so the key point here is, again, this underlying principle of control, and hopefully that will cross-cut everything I'm going to show about today. And I've mentioned this. So the model I'm going to talk about today is salmonella. Most of the work is going to be salmonella in a mouse, but there will also be salmonella work using human material, and then one slide on COVID to sort of show how these studies bridge. So if you have a salmonella gut infection and it stays there, you've got an incredibly low risk of death. If that bug, the same bug, just goes through the barrier and spreads throughout the host, and the bacteremia here is one organism per mil of blood, it's tiny. And that's enough to change your risk of death for typhoid to untreated typhoid is about 1 to 10%. For untreated invasive non-typhoidal salmonella infections, it's about 1 in 4. These are staggering numbers when you think about it. And yet it's the same organism, and only its locational distribution has altered. And so there's a rich history of salmonella infections being associated with cardiovascular and thrombotic complications. The oldest paper I've been able to find is about 120 years ago, old or so. But there's a whole range of these, but it's not really thought of as a byproduct of having a salmonella infection. It's just the presentation and how it appears. But actually, even if we focus in on non-typhoidal salmonella infections, those ones that stay in the gut, then studies suggest is that you're still left with a significantly increased risk of one or more of DVT, pulmonary embolism, and stroke years afterwards. Okay. So on top of these can have multiple presentations. I'm largely going to focus on venous thrombosis, but these can result in organ failure and damage. And remember, even in that worst case scenario I gave you about invasive angio, 75% of those individuals are likely to survive in that instance. Now, that means is that there's the potential for long-term damage to be inflicted on the host. And the impact of salmonella infections in the mouse and in the human are really quick, at least on platelet numbers. You can see here, so mice have more platelets than humans, about 3 or 4-fold more. And then, but within a day, you can see is that there's a rough halving of the numbers of platelets, reaches its nadir between this day 7 and 21 period before it recovers in this recoverable model. And then you can see a reciprocal change in mean platelet volume. So the impacts of salmonella infections, like you'll see in very many serious infections, is rapid. And because this stuff was published a few years ago, I just want to focus on what we originally found. And we found is that given infection drives a thrombosis in the liver that we detect at day 7 after infection. We don't see it at day 6, we don't see it at day 5, but we do see it at day 7, okay? And that's important for what I'll show. When we home in on what these thrombi look like, and they can really block the vessel all the way through, this is the portal vein, is that the thrombi attach, you can see how the platelets here in blue go under the surface endothelium and can make contact with parenchymal sites. The thrombi themselves are white, which means platelet rich, but red cell poor. They contain this classic fiber and capricate you'll find from Willibum fragile, a whole range of different things. And the point here is that they appear at the same time as you see organized, I'm going to call them for simplicity granulomas, they're inflammatory foci, but I'm going to call them granulomas for the purposes of the talk. And here you can see these granulomas are organized structures containing salmonella in the green dot here, a tiny number of salmonella for a big granuloma, macrophages, F480, positive cells, neutrophils here in red, which are in Ly6G. You can see the INOS-rich sites and so on and so forth. And you can basically contain a myriad of different cell types as well. And so this strongly suggested to us when we were looking at this is that the inflammatory response and the thrombosis were tightly linked. And so here we've got vessel occlusion. We can see is that there's, I haven't got 0 to 6, but nothing before here. At day 7, day 21, you see it's long lasting. So for 3 weeks, you've got a persisting thrombosis response in the organ, and it tails off after about a month or so. And those granulomas, you can pretty much superimpose the kinetics on there. And in contrast, if you look at bug numbers, they reach their peak at day 7, which is when you find here, but when you've still got quite a strong thrombosis present, bacterial numbers now are less than 10% or 5% or so of their peak numbers, and then fade afterwards. And it's got nothing to do at all with bacterial numbers in the blood, because these are pretty much undetectable in this model by these later time points. Okay. So other evidence that these were linked. So this was a really good link between the two, but we went and showed a very simple way, is that we infected TLR4 knockout mice, which TLR4 is an immune sensor for LPS, amongst other things. When you infect these mice, they look like non-infected. You don't form granulomas, and then you don't see thrombi in the vessels. And then actually there's a relationship between the TLR4 ligation and the amount of interferon gamma you detect. So it's a really tightly linked relationship here. But in many ways, we need to be careful about what we wish for. And the reason for that is that if we now take a step back and look in other inflammatory mediated models, so here, for instance, we're looking at wild-type mice and interferon gamma knockout mice. Now, these mice have more bacteria at this day 7 time point than wild-type mice. If we look in the which looks like the TLR4 knockout, and if we look in the vessels, we in the liver, we pretty much see 0 thrombi in there. So this again delinks further this relationship between the need to have the organism, the inflammatory response, and the level of thrombosis. If we now look in another knockout mouse, and this is a mouse which lacks both TNF receptors on their surface, is that you find is that the bacterial numbers at this day 7 time point are pretty similar, sometimes up, but roughly the same. And then if you look in the tissues a bit more deeply, you get inflammatory foci and accumulation of inflammatory cells. They're obviously a little bit less organized because TNF plays a key role in that. But now you've got same numbers of bugs, inflammatory foci, but no thrombosis. So it tells us is that although inflammation cross-cuts both the development of the granulomas and thrombus formation, that actually you can decouple this in some ways as well. And finally, what if you take the brakes off? So what if you take interleukin 10 away and from here? And actually, mostly these mice do better. They have lower bacterial numbers in their tissues at this time. They also make more granulomas. And lastly, they have really, really extensive thrombi all the way through the tissues. And in fact, this is called low dose for a very simple reason, is that we have to lower the dose to be able to not kill these mice with this infection, which is reasonably well tolerated by or survivable by the wild-type mice. So here now, if we take a step back and think about it, if we go all out to control bacterial numbers, that's great, we can easily do that. But think about that nuclear weapon, and then here, there's a price to pay for success. And I'm going to cut to because all this is published. So what we found was that salmonella infection triggers TLR4, results in interferon gamma. You get an accumulation of monocytic cells, amongst others, protoplanin expression on these, collecturon platelets, and leading to thrombosis. And we were able to demonstrate this through a whole range of different knockouts and so on and so forth. But there was a problem here, and the problem that was bothering us was that we knew this was a systemic infection. We knew if we looked in sites like the spleen, is that we had roughly the same, maybe even slightly higher bacterial numbers. But at this day 7 time point is that we really would see very, very few thrombi in that organ. So we started to think well, maybe had we missed it because there are so many immune cells in the spleen, maybe we looked at this incorrectly. And Noni was brilliant at really sorting this out. And what she did is that she took a step back and said, well, let's look really early. And so she looked from 8 hours. I'm showing you from 24 hours here, but she looked from 8 hours and so on. And what she found was that actually, if you looked at the spleen in a slightly different way as well, is that at this extremely fast time point, is that the spleen was riddled with thrombi, and you can see this just zoomed in here, in the splenic vein here all the way through. Okay. Then putting into a figure a huge amount of data, what it shows is that to the single infection, is that the induction of thrombosis was distinct in terms of its kinetics in each individual organ. So in the spleen, you could detect it super quick, but by 24 hours, it would largely resolved and then bounced along the bottom. This probably over-exaggerates what we're seeing. Bounced along the bottom here. But the key point here is that it means it can be extremely quickly, rapidly induced, but also extremely rapidly resolved. And this has implications for stability of thrombus, downstream sequelae, etc., and so on. In contrast, the liver, once it's up, it's largely up. It stays there for weeks afterwards. So there must be at least some distinct features of this response in different organs to the same pathogen. And in terms of sepsis and consequences of sepsis, I think this is incredibly important. So there's predictable and reproducible rounds of thrombosis in a single host to the same pathogen. All right, so if we just, I'm not going to go into this in much detail. All it is that if you look at a thrombus from the spleen and then the liver, they're largely the same. They're platelet rich, they have lots of neutrophils, they have some inflammatory cells, there's plenty of fibrin in there, and then this is just what if you put these all together. Okay. But there's something missing from here, from the story, and that is where is the pathogen? And so it's really a bit player in this story, because one of the things that might have been occurring is that all those bacteria were getting trapped in a thrombus. So this is inside a vessel here where we can see it flanked, and the red here is picking up on platelets, and there's our single organism here. And I love this figure so much. And then, so if we, and Marisol was particularly fantastic at doing this, is that what she did is that she took many thrombi, cut through them each time, looked at them, and did a count for the frequency that you could detect bacteria. And essentially, most of the sections that were cut had 0 bacteria associated inside the thrombus, and about 20% had 1 or more. And then from that's about 50% or 60% of these only had 1. So the presence of in fact, that's what that shows. So the bacterial infection was triggering this process, but they were bystanders pretty much thereafter. And so we can look at this in a slightly different way, and this is probably important for what we think is happening. Although you don't find the bacteria within the thrombi, you quite often will find bacteria proximal to the vessels where the thrombi are formed. Now, it's very difficult doing a statistical analysis of this, but it's led us to hypothesize that that's probably the trigger, and this is the crash site, and this is the airbag going off. Okay. And quite nicely, I just like this. This really shows how it's a very thick section of this, with the thrombus here in red, the vessel wall here in blue, and there are 3 bacteria, which if you just look at the vessel, it looks like they're inside, but actually they're on the outside here of this vessel. So if the kinetics of thrombosis are different, then are the mechanisms between the liver and the spleen conserved or different? And so one of the ways that we did this is that we cleared out, we knew from what we'd shown before in the first papers that monocytes were essential for this, or clogenate sensitive cells rather. And then if you knock out clogenate, or if you collagenate treat, wipe out your macrophages, you basically lose all your thrombi formation. So that was conserved. So you need that cell type. If you now break this down to, do you need interferon gamma? And in the liver, it was an essential component. In the spleen, it's probably contributing because of the stats say it is, but it's certainly not essential for this, because you see plenty of thrombi forming and decent-sized thrombi forming, irrespective of the presence of interferon gamma. And also, probably really quite surprising to us, and this was this terminal interaction between podoplanin and CLEC2. If you remove CLEC2 from platelets, and then you infect, and then you still see that there's probably a partial effect in the spleen, but it's certainly no more than that. Okay, so that means you've got both a conserved backbone and some variation based on the simply based on the site where the thrombosis is occurring. Okay, so what about neutrophils? So neutrophils, as we can see here, so this is a whole liver section here shown at scale, and you can see the thrombi form within here and stain for neutrophils and fibrinogen, and this sort of like dirty brown color is because the thrombi are extremely rich in both of these factors. Neutrophils are present throughout, and fibrinogen is found within the thrombi as well. Moreover, if you start looking in the spleen or in the liver as to who captures the cells, is that you find that the bacteria are much more likely to be found in a macrophage-like cell or whatever, both in the spleen and in the liver, than you will find them in a neutrophil. And this probably overstates, actually, because this is done by pixel counting, is that when you do it by eye and by hand, you probably find about 3%, 2% of bacteria are found associated with neutrophils, whereas about 95% plus are associated with macrophages. So what this means is that the neutrophils are contributing to this process, but not because they're capturing bacteria. Okay, so what did we do? We do what everyone else does, and we knock them out. And so you see here, both in the spleen and the liver, and this is published in a recent paper with Marisol's first author, is that if you want the details on how we do this, but in short, if you remove neutrophils, and it doesn't even have to be a complete ablation, is that you of neutrophils, is that you remove the opportunity for thrombi to form. Okay. So now we know the cell types are definitely contributing to this massively, and we can look at this. So this led us then to start really thinking, both in terms of the immunohistology and everything else that we were considering, is how does this really look in the vessels? You know, so we performed intravital imaging of post-infection. So a little bit of a explanation as to what we're looking at here. So platelets here are shown in red. Neutrophils are here in blue, and CD11B here in green. And so this is an infection which is being imaged 6 hours after an infection, and the recording has been made over the period of 1 hour. So what you're going to look at in this video is a 1 hour in this animal's existence. Okay, and you can see here is that although there's a little bit of movement, you've got neutrophils which are already established within the vasculature. You can see here that they're probably at a higher concentration here. There's some interaction occurring with platelets in the vessel, but that actually mostly the platelets still remain free flowing in this at this time point. So you can see this probably thrombus formation starting, but it's not absolutely complete. This other parts here are within the spleen itself, of the red pulp of the spleen. Okay, so this certainly supports the concept that what we're looking at a effect in the vasculature for neutrophils. And if we now take this to the next level, and this is a 15-minute recording taken from a mouse which was infected 24 hours before. So this is now when the infection is extremely well established. And now we can see that the thrombi are well-formed platelets move with much slower dynamics and kinetics through the vasculature, that there's, again, a lot of ongoing interaction between the two different cell types here within the vessel, but to a much lesser degree within the parenchymal sites. Right. So how are neutrophils contributing to this? Well, the obvious way would be through nets, neutrophil extracellular traps, and it is without doubt that we observe the formation and of nets in this response. So they're obviously likely to contribute, but if we use a classical approach, which is to add DNAs to the mouse, at least in the experiments that we've done, there's at best a partial effect on this. In some mice respond, some mice don't, but that there's not full activity. So we think probably nets are contributing to this process, but we don't think necessarily that they are the only answer here. Okay. So what could be going on? And I'm only really going to summarize aspects of this data because there's a huge amount of it. It's largely conserved between both sites, but the reasons are probably slightly different. So I'm just going to, and also once you've got the process rolling, is that the downstream effects are probably conserved between, in the basic sense, they're conserved between both sides. So if we take monocytes and neutrophils and infect them in vitro with different levels of bacteria, we find that many of the monocytes that we have in vitro produce TNF, whereas only a marginal proportion or so, it could be assay development, produce TNF after infection. But the big take-home message here is that both cell types can produce TNF. So if you now neutralize TNF, as I showed you before, is that in the liver, is that you've got no thrombosis, but it's also the same in the spleen. So you've got a near complete dependence of complete dependence on TNF for this process to occur. So what does TNF do in this context? Well, it looks likely, but if in the same neutralizing experiment, is that TNF is involved in the endothelial activation. This is measured by looking at CD62E distribution along the vasculature. And then furthermore, is that there's also, if you look at the anti-PSGL1 activity, if you endothelial activation can lead to PSGL1. This leads then to, if you neutralize this essentially it helps home cells into the tissues. If you neutralize this, you impair impact the level of thrombosis detected. Interestingly, in nearly every instance, is that we see it's harder to stop thrombosis in the spleen than it is in the liver. Okay, so that's what happens in the absence of any adaptive response in the mouse that or the necessity for any adaptive response in the mouse. And so we're looking at this slightly differently now in human platelets. Okay, do we see something like this with salmonella mixed with human platelets? And like, if you bang two rocks together, if you mix salmonella with platelets, washed platelets, whether they're mouse or human, you don't see any activation. It's a fairly clean result that nothing's happening. But you can make this happen. So this is agrometry, I hate that word, And what it basically means is the amount of, it's a classic way for detecting platelet aggregation, and it basically is about light transmission. And so if you take human platelet-rich plasma and then you mix it with bacteria, what you find is there's a huge spread in the ability of donors to be able to lead to platelet aggregation. It's a huge spectrum altogether. But many can to some degree, and that also links into the lag of this. Oh, sorry, I seem to have reproduced the same thing twice. Okay. So to cut a long story short, is that what we hypothesized and turned out to be correct is that this would correlate to the amount of specific anti-salmonella antibody that was found within that plasma, within those individuals. And then, so what we were able to do that through a range of different ways, I'm only showing one of the ways here, is that if you depleted the specific antibodies from that plasma, is that then you lose the ability to aggregate platelets. Secondly, if you blocked the ability of human IgG to bind to platelets or interact with platelets through Fc gamma receptor 2A, then you also lost the ability to aggregate platelets. So what this led us to conclude is that IgG binding to salmonella and Fc gamma receptor 2A is required for human platelet activation in vitro. Now, it must be said that the inflammatory cascade type responses I showed you for the mouse are just not possible to be performed in humans in a simple way. So these studies really, really help us in this context. Okay. So, is this relevant for human infections? The short answer is yes. This figure is taken from Andy Pollard's typhoid human challenge model. And in those individuals who got sick, if you followed them and looked at day 14, when we know that thrombosis is still active in the liver, is that these guys have an increase in their in their specific anti-salmonella IgG responses. I think this is LPS, yeah, so LPS. So what we think is happening here is that salmonella plus human platelets, if there's no antibody and you don't see aggregation, so there's no basically cross-linking here. If there is antibody present, is that it leads to the aggregation of platelets, and this is entirely dependent upon Fc gamma receptor 2A. It's a big picture type approach, this, is that it says is that it's possible to achieve something here. We're probably missing out on some mechanisms at play. Right, so what does this mean now for in the context of humans? So the first thing is that the thrombi that we observe in the mouse are virtually identical to what we see in the humans, and this is image, a couple of images here from a study which was published during COVID, and we were shocked. They even have the same shape and the same look, and we confirm this in other ways. So the pathway that we think we've identified for to drive thrombosis in the mouse, we think is highly relevant to what happens in humans. The second way is if we take antibody-mediated effects here, and if we look at COVID in another way, is that individuals with severe COVID are much more likely to have antibodies against this intercellular cement molecule, desmoglein 2, and then individuals who have been hospitalized and in ITU for other reasons, or who've had flu, for instance. And in fact, they match patients who have had some presentations of cardiovascular complications here. And then lastly, how does this matter? Is that if we've got cardiac tissue from postmortems from individuals who did not die from COVID or who did die from COVID, and they've been labeled with an anti-GSD2 antibody to pick up desmoglein 2. And basically, this you can see here is that they're thicker dark bands throughout this tissue, if you look in PM from COVID, this separation, which you can see here, you can see them laddering, is indicative that there's a concern that there has been a separation between these two bands, so that the tight junctions are a bit weaker. And lastly, is that if you now look in just label for anti-immunoglobulin, so what we're really picking up here is are there anti-DSG2 human antibodies from the host already deposited on this? And then the short answer is yes, you can detect it after COVID. So these aspects of this combine, and hopefully they'll be expanded on in the next step. So just to summarize, it's the complexity and the regulation and all of this relates to its importance. It's to prevent organ and cardiovascular damage is basically the upshot. I'm not going to go through this for reasons of time. But thrombosis develops in different organs with distinct and, incredibly importantly, predictable kinetics to a single pathogen. There's a common mechanistic backbone, but each site has its own organ-specific features. We hypothesize that the trigger is bacteria being proximal to the vessel walls, which zip codes or postcodes wherever you're from, where thrombi form. We also hypothesize that macrophages play a bigger role in tissues in this process, whereas neutrophils' role is more restricted to the blood. Few bacteria found in there. We've evidence that the human IgG is to O antigen, and this drives human platelet aggregation to Fc gamma receptor. And what we think is that when you have chronic infections, so lots of antigen, at a time when IgG is formed, this may cause thrombosis or at least immunomodulate or modulate in some way. And then bacterial killing, here what we're questioning is whether when we're looking to control infection, how much do we need to focus on bacterial killing versus restricting antigen access to organs? And I'll stop there and thank everyone. I thank the major people. This is obviously a huge team effort. I'm lucky enough just to be the spokesperson, really. Thank you.
Muchas gracias. Thanks a lot. Pueden hacer sus preguntas también en español. La doctora Cristina va a coordinar la sesión de preguntas y ella puede traducir esto. La doctora Cristina Gil, ella es egresada de la Facultad de Química de la UNAM, hizo su maestría y doctorado en el Instituto Politécnico Nacional, estuvo en estancia en el laboratorio del doctor Adam Cunningham y ahí publicó un artículo muy importante en PNAS, que abrió una brecha muy importante en las investigaciones de vacunas contra la salmonelosis no tifoidea, y después migró al laboratorio del doctor Burkhard Ludewig, en donde ha desarrollado importantes avances que pronto serán presentados, también parte de ello, y ha ganado varios premios de Syntex y ha publicado en revistas de las más importantes del planeta. So the session is open for questions, guys. Yeah, our students from Egypt. Yeah, you can. The floor is open, and so. So while you're thinking about, oh yeah, okay, thank you.
Hi. Hi, thank you very much for this great talk, Dr. Cunningham. Very nice to have you here in the National Academy of Medicine. And I know that you are very focused right now about the Salmonella and their effects in this thrombosis related with inflammation, but this is just particular of this kind of bacteria, or we can also expect that other pathogens such as virus or even some parasites could also have this kind of increase endothelial coagulation system overactivation and all that?
Yeah, great question. I I think so. So the reason salmonella is, so I use salmonella for everything, pretty much. The reason it's so important in this context is because you can control the outcome, and by that I mean the survivability of the mouse post-infection. So you can look at it without having to have. You don't need too many bacteria to infect, and you can look over time. So what we think here is that that's exactly what it reflects, is a general principle that can occur. Some of the molecules say, if for salmonella you could swap in E. coli, you could swap in Klebs, you could swap in any gram-negative, because obviously they've got a little bit more there, particularly with that TLR4 conserved. Whether you then are able, for gram-positives, they're probably a little bit easier. So there's a structural component here on the bacterial side, is that bacteria spend a lot of their effort trying to not be seen. And for salmonella, that has a very long O antigen chain. So it means is that to see those, surface molecule, really super proximal surface molecules, just got to try a little bit harder to do that. That's maybe not as the case, for instance, with gram positives, where you've got a greater dependence on the capsule for to hide from the world, compared to you don't have long O chain LPS. But the principles are certainly the in vitro human stuff, is that the evidence is that many gram positives are super quick at causing human platelet aggregation in the presence of antibody. So I think that's the case there. The other thing I should point out is that, remember, nominally, all this is happening with salmonella, which is a nominal intracellular pathogen. So this comes back to, once you kickstart a process, then how much of this is a failure to control, and how much of this is because of exposure to bacterial antigens. There's probably somewhere between the two, obviously, because no infection, no thrombosis. But you do need, it's really, this is about a particular failure in control to each of these processes. In terms of parasites, again, great question. I suspect, whether we know many infections, but parasitic infections, malaria, will have these types of effects. It's extremely well documented, but the reasons why aren't, and that's what I think this brings to the table. Viruses, they're really small, so they're outside of I can't be bothered with them, if I'm perfectly honest, in the main, because they're really hard to model. I find them, COVID in humans suggests that this is a conserved process. Many of the same factors, are present, but there may be more variation for individual viruses.
Thank you, Professor Arriaga. Professor Ludovic?
Yeah, you may call me Burkhardt. We have been friends for so many years. Professor, thank you. Adam, great talk, fantastic. I mean, it is very nicely delineated what appears to be the major conundrum. Why so much fuss if there is only one or sometimes even none of bacteria in the thrombi? Could it be that it's either an overshoot, that there is the initiation still dealing, right? The initial thrombus still dealing with more bacteria, and that that could be one possibility, or you mentioned it before, is that there is a lot of antigen, because you need the antibody, you need the antigen, right? And then, of course, the platelet. So is there the possibility that there's still a lot of antigen around, and that this is then determining also the delay in the overall thrombotic kind of overshooting inflammatory response?
I can deal with the second part first, which is there's one thing I possibly omitted from saying, is that human platelets express Fc gamma, the low affinity, but mouse platelets don't, which is why I was able to say this is in the absence of IgG driving this. But I think there is an antigen. Ironically, we don't, when we've done a lot of studies, a lot of experiments, staining for bacteria, and we know in some context, ironically, when we don't see thrombosis, is that we can see a huge amount of antigen when the recoverable bacterial numbers, colony-forming units, is extraordinarily low. So that would argue against that this is just simply antigen depot. There may be some minutiae within that the specific antigens aren't picked up by the screening sera that we look at. We know that porins, for instance, are probably if not the first, and certainly in the first couple of antigens which were ever identified to be able to interact with the thrombotic cascade in this way. About 40 years ago. Going back then, if it's an overshoot, again, using sampling beyond just intravital, we've never seen any evidence of that. In videos I didn't show, you can see if you introduce the bacteria at time point 0, they nearly always get mopped up by macrophages. It's just always the case. There's nothing that's happening. The CFU counts in the blood are really low. So we don't really know if it's a pathological consequences. It's conserved across many, as I answered in the last question. The ability to drive a thrombotic response is very heavily conserved across different pathogens. But it looks likely that trade-off between it being beneficial, immunothrombosis versus inflammatory thrombosis, is probably where this comes in, and this may be influenced by dose, localization, etc.
Christina? Thank you.
Thank you very much for the great presentation. I have a question about the kinetics of the generation of the thrombi and also the granuloma and the clearance. Why do you think that these kinetics are so different? Is it something to be, because the bacteria spreads across the organism, so you have bacteria in both the spleen and the liver. So do you think it is something to have with the cells that are sensing the bacteria in that place? You show that the bacteria is mainly kept within macrophages, and the macrophages in the spleen and in the liver are very different. Actually, it had been shown that the macrophages in the liver are like firewall against spreading of LPS. So do you think that is something related with the cells, the kinetics of the bacteria, and why they stay longer in one place than in the other?
I think that most definitely is likely to contribute the type of cells which are present. So we've phenotyped the monocytic lineage cells, if you want to call them that, in the liver in really quite great depth in the original paper. So the upshot is that at time point 0 is that you've largely got Kupfers, as you'd expect, maybe a couple of others. But within 24 hours, you've got inflammatory monocytes coming in. By day 7, you've got a really heterogeneous phenotypic presentation of cells, monocytic lineage cells, which are probably a mix of different types. And then how they contribute. So the short answer is I don't know. And thrombosis to anything is common, even lying down, but it's just a very different mechanism. The few surprises is that this wouldn't have been expected to be seen in the venous circulation normally, a white thrombi. So what I think is possibly happening is that the changes in the cell type which are within the tissue, alongside changes in the platelets which are found within tissues, makes a huge amount of difference. If we take the very simple view, that is it on a is it at a numerical basis, then that's not likely to be important, because when it crashes in the spleen, you've still got cell numbers going up. You know, you're still getting an accumulation of cells in the spleen. So it is something about those. I don't know what that is. In fact, the rate of involution isn't very well explored or understood, particularly in pathological models. What I do think is that maybe taking a step back is that we should be thinking about this in terms of risk. And you've highlighted the bone marrow there. We know that you get a diminution in the numbers of megakaryocytes in the bone marrow immediately after infection. You get a megakaryopoiesis occurring in the spleen, and then it switches into the liver at a later time point during the infection. So it's constantly changing, which may affect the type and quality of platelets that you're producing until it goes back in. But it still doesn't explain why the difference in the spleen and in the liver. So that's probably a really, really long-winded way of me saying I don't know. But I think the important thing is that by having something to understand the risk and when the risk is that possibly that's, you know, you've probably got different phases of this. Early risk, late risk, when things go back to normal, inverted commas, in the bone marrow.
Thank you very much.
Just what will happen if you have vaccinated animals, like with OMVs or porins, and then you infect them? Will they make the thrombi? What will be the antigens that are triggering this thrombi-related response? Would be LPS or something else?
LPS is the go-to antigen that everyone uses for these types of studies. So LPS can do this, but if you just look at the sheer numbers of LPS molecules which are introduced into a mouse to drive this, it bears no relationship to even the most staggeringly widespread infection that you see. I think, again, you need some organization on the presentation of antigens for this. If you challenge a vaccinated animal, I don't think I've ever seen a thrombus after challenge, which is, again, a really important point, is that vaccination can save lives even through these processes. In fact, flu vaccination drops your risk of cardiovascular events as well. I think the introduction of any antigen probably can drive a thrombosis, but because it's not it's a very short-lived, probably clinically, subclinical presentation of this. But,
yeah, and the role of this marginal zone macrophages. I mean, have you dissect more on the spleen? With which populations of macrophages?
Fantastic question. No, we haven't. We know that they can capture bacteria, so that may be a triggering factor. But so the other thing I didn't show in the interest of time was that you get changes in platelet leukocyte aggregates as well. So platelets are often piggybacking on other cell types, and so you can get changes in there. And to be honest, offhand, I can't remember what it showed for the marginal zone, but we haven't been able to break it down to that level of resolution. It's a really important point.
Maybe with the osteopetrotic animals, they lack.
Sorry?
The osteopetrotic mice, they lack the marginal zone, and maybe that could be a model. Yeah, and what you observed, I remember the rosella, the typhi rosella, the rose spots. I mean, that could be one of the mechanisms that we.
Yeah, it could be. I'm not sure if they've ever been dissected in that way, but I can say is that if you look at the intestinal perforation, there's a strong association with thrombosis around that point, and that's associated with the greatest likelihood of poor outcome in typhoid. And you can see that there's. I think the problem is I'm not sure Burkhard had a comment very similar. I think you both did yesterday as well. Is that people say these things and they report, oh yeah, there's a thrombus or there's a heart damage, but getting good quality data to show this at a population scale it's a lot rarer. It just shows what we need.
Excellent, and thank you for this great talk. And Dr. Christina Hill will give you
Thank you. Thank you very much.
And continue with the program. Let me introduce to Christian Perez-Shibayama. He is also a QFB, a chemist from the Faculty of Chemistry of the National University of Mexico. And he did also a master's and a PhD on immunology. And then he moved to Switzerland with Professor Burke-Ludewig, and he has achieved a lot of fantastic results on his research and has been published in very important journals. So thanks, Christian, and please introduce Professor Ludewig.
Good morning, everyone. As Constantino said, my name is Christian Perez-Shibayama, and it's a great pleasure, a great honor to introduce such a distinguished speaker, Professor Burke-Ludewig. Thank you very much. Professor Ludewig is the head of the Institute of Immunobiology in the Kantonsspital St. Gallen in Switzerland. He's also the head of the cardioimmunology research at the University Hospital in Zurich. He studied veterinary medicine in the University of Berlin, right? And obtained his doctorate degree in 1995, but then he continued his postdoctoral training in different places, Robert Koch Institute, the Hebrew University of Jerusalem, and very importantly, in the University Hospital in Zurich at the Rolf Zinkernagel Lab, which is a Nobel Prize in medicine. And it was the time when he met actually Constantino at that moment and starting a long-lasting collaboration with Mexican immunology. So it's a great pleasure to have her here. He's a pioneer in not only in stromal cell immunology, but also about cardioimmunology, and he's recognized as a global leader in this field. And Burkhard, it's a pleasure to have you here. I'm very much looking forward to your presentation.
Yeah, thank you very much, Christian, for the very kind introduction. You still can call me Burkhard, right? I know it's very formal here. But Constantino and Christina also, thank you very much for the invitation, and it's great to see you all here. I will, as mentioned, talk about cardiovascular projects that we have been pursuing over the last years, and it's about T cells, as indicated in the title, and I would say the extended title, or beyond the subtext, is that myocardial inflammation, it's immunology and beyond. So what are we talking about? And thank you, Adam, for introducing some of the complementarity. I will focus a bit more on the translational aspects of a particular disease. And since the audience is full of young researchers, young people, I think you might be in particular interested, because I will talk about myocarditis, which is worldwide, mainly Europe or in the Northern Hemisphere, a disease of young adults. So this is a case that a young lady who is in our study that we're following the disease. She was recruited into the study, and we're following her. She had, before she was recruited in 22, a post-COVID associated perimyocarditis, so a global inflammation in the heart. Well, there was pericardial effusion, so that's why it is pericarditis. She then recovered, probably did not think that it's something serious, as many cardiologists in these days still think, at least in Europe and Northern America. But after last year's very severe flu, epidemic, she contracted the same disease symptoms again, chest pain, very similar to the previous episode. She was then admitted to the cantonal hospital in Lucerne in central Switzerland. The ejection fraction, so the pumping function of the heart, dropped dramatically. Normal is everything above 50 to 60% of the percentage of the fraction of the heart that is pumped out of your heart with every beat. So she received an implant, which is a little pump into the left heart that further helps the blood to be pushed out of the left heart. But she then went, that did not help that much, she went into cardiogenic shock, was then transferred to the University Hospital Zurich, received, in addition to the impeller, to the little pump, an extracorporeal membrane oxygenation, because heart function was so low, so the blood had to be oxygenized. Still, there was no clear diagnosis. So what they do then, the physicians, they just give a bolus of prednisone. One day later, the endomyocardial biopsy, so which is via the jugular vein, a little catheter introduced, and then some small tweezers, forceps are inserted, and a very small piece of the heart wall in the right heart is taken. That showed CD3 positive T cells and macrophages, so that were established the diagnosis. Currently, what is done in these severe, or even slightly less severe cases, cardiologists throw everything on and add everything. So anti-IL-1 therapy, anakinra, and even intravenous immunoglobulin. Sometimes a very old method. It's called a anti-thymocyte globulin, which is in essence a polyclonal serum generated in rabbits to deplete lymphocytes in humans, and it's still given, and that's roughly what is out there for this disease. And luckily, it helped here many times before. So you realize in this disease, intensive care is very important. Keep the patient alive, and then do something that tunes down, right, that follows the theme that we had before, something that tunes down overshooting immune reactivity without knowing how it exactly works. And that's, in essence, our mission. I'll summarize very briefly how we see myocarditis. In these days, we are more confident that this is true, and it's a simplification, of course, but immune checkpoint inhibitor treatment associated myocardial inflammation just shows us it's the T cells. So, and how do T cells, and I have been informed it's a mixed audience, so we are in between cardiology and immunology, so sometimes it's a bit simple, sometimes it might be a bit too complicated, so forgive me for that. So the simple view for the pathogenesis of the disease is that it's either, and we heard that before, it's the same theme, either it's the brakes off, for example, immune checkpoint inhibitors, then T cells directed against the tissue are switched on, so they are activated. It's CD4 T cells and CD8 T cells, or there is an accelerator function. This can be either molecular mimicry, because bacteria present similar structures that activate cross-reactive T cells. It could be any viruses. It does not need to be a virus that replicates or damages cardiomyocytes or replicates even in the heart. It can be any global immune activation, and of course it has been discussed as part of immune activation, and it's shown that part of the immune activation associated with vaccines, with any vaccines, can push the accelerator on the T cell activity, and that leads to the disease. How is it working in general? Again, for those who are not familiar with immunology, that might be a bit too much. For immunologists, might be too simple, but I'll try to go through that here. So there is, for T cells in particular, they have to go to school. The school for the T cells is the thymus. When they leave the thymus and they pass all the exams there, they're called naive T cells. They get the further education and the instruction right? You use the military equivalents, right? So the army has to be instructed, and the instruction of the army and the expansion of the army, the basic training and expansion can be our lymph nodes. Then the T cells are called effector cells once they have received instructions, and they go to the target tissue. And here for myocarditis, so inflammation of the heart, into the heart. So how does the education in the thymus work? What is important is that antigens, so structures that are recognized by the T cells, are either present or not present in the thymus. If they are not present in the thymus, so such as the immunodominant antigen in this disease, the myosin heavy chain 6, or the heavy chain, alpha heavy chain of the myosin, it's not in the thymus, and hence one part of the test in the thymus is, do you see cells? And if the answer is no, then these T cells can leave, and this is what's happening in the thymus for that particular antigen. That's why it is an immunodominant antigen, because it is not presented in the thymus. So there's the possibility that these cells are activated in the lymph nodes by, for example, dendritic cells or other professional antigen-presenting cells. If these T cells see and are instructed by this to react against this antigen, the myosin heavy chain 6. Again, here, for example, for the CD4 T cells, and then there are activation pathway circuits that lead to the expansion of these cells, and then they can go into the heart. How do T cells damage the target structure? So the heart here, it's either the CD4 T cells. They are not killers. They do not directly kill. They have their hitmen, and the hitmen of the T cells are the inflammatory macrophages that are activated, for example, by interferon gamma and other factors, and then they secrete factors that can damage the target tissue. Of course, not only eliminate bacteria, in particular intracellular bacteria, but it's a very similar mechanism. And CD8 T cells, they directly kill the target cell. So again, in myocarditis, it's again same theme as if we had talked about that before, but that's how immunology works. It's either to regulate, right, to regulate in a sense, to eliminate a damage or a pathogen, a threat, a perturbation, and at the same time to keep tissue integrity and to maintain tissue integrity. And this is disturbed if, for example, there's molecular mimicry, cross-reactivity. There is this balance is disturbed if there is excessive immune activation once T cells or antibodies are produced in a misdirected or produced in an excessive fashion, or what has been mentioned before, the equivalent is we've heard about that IL-10 is if IL-10 is not present, then there is a lack of regulation, and then there's excessive tissue damage. No, that's that's a lot to take in for the cardiologists. I don't know whether cardiologists are here. So when I present that at cardiology meetings, this is wow, right? This is a lot. For immunologists, this is of course our paradigm. But important, as Adam has mentioned, it is important for us. It is very important. Has been important to follow and to assess whether and to what extent these principles are really operational in the patient. So myocarditis in the patient, there are different, and it's an interesting disease. Why it's an interesting disease? It's a disease of young individuals. So the patients in our cohort see a hospital many times, first time in their life from the inside. So they have been hit first time. Second, it's interesting because there's quite a variability in the disease. So young individuals never seen anything before, and there is a variability from milder course and to a fulminant, the fulminant case I've highlighted. If in these days, because intensive care is so efficient and immunosuppression is present and there's more heightened awareness for the disease, if immunosuppression and maintenance of cardiac function is guaranteed, then there is a post-episode appearance. There's either resolution, there is recurrent, as in the young lady we've had, or a still more theoretical scenario that is chronic. It's just persisting. Or right in infection immunity, we know that as a persistent antigen, persistent pathogen-driven chronicity. How to study that, and I would also like to mention Christina, because it was Christina and myself 5 years ago who went to Zurich and set up this study, which is now supervised and as the clinical principal investigator led by Dörte Schmid, and 3 medical PhD students who are working with us, recruiting patients with acute and chronic myocarditis. The clinical definition has been, I spare you that, but the confirmation that it is indeed myocarditis is done by either cardiac magnetic resonance, so CMR, and or endomyocardial biopsies. And we focus today only on the variation on the disease presentation at presentation, so at day 0. We follow those patients for 3 and 12 months. So the recruitment, it's a rare disease. It's also, in terms of medical terms, it's an orphan disease. So there is no dedicated and cause-specific therapy. Overall, we have recruited until last month more than 100 patients. Overall, I'll summarize here. At baseline from Zurich and from St. Gallen, overall it's 79 patients, and from the acute myocarditis, 56. We even have already a 3-month follow-up. I'm presenting here, and it's always an intermediate summary. So in our study in pancreatitis, I present data from 65 patients. I've mentioned that before, so the median age is 28, between 17 and 70. We are we're recruiting. After we know the demographics of our indicator of our patient cohort, we recruit selectively healthy controls. So same median age, same spread. As mentioned before, the majority of the patients is male, as in the controls. All of them are diagnosed positive by cardiac MR, and the left ventricular ejection fraction, or EF, I would call that in the following, is also varying, median 53, but some of the patients have a very low function of the heart. In clinical research, what do you need in addition? Of course, you have your main cohort. You always need a validation cohort. We're lucky to collaborate with the University Hospital in Tübingen, and they sent material, and we could explore the patient data from another 22 patients, so that the overall myocarditis cohort, the data I'll present, is 87. Healthy controls is still the same. What you can appreciate here is that the fraction of females is increased, and what is also changing and introduced, the change in the cohort is because the patients in Tübingen showed a much more severe phenotype. They were all from the ICU. But all of that, I spare you more details. That confirms, in the validation cohort, confirms what we conclude. Summarizing here the data, the clinical data from the aggregated cohorts from the 87 patients, always in black the healthy individuals. And what you can see here is this is C-reactive protein, so the reaction of the liver to a disease in terms of inflammation. So there's a significant increase. Damage in the heart, indicated by troponin I, can be detected in almost all patients. NT-proBNP, or only B natriuretic peptide, is a very important regulator that is immediately released and produced by the heart and by the kidney at a situation of cardiac damage and volume changes, so requiring adaptation of the heart function. And as indicated before, one measure for the heart function is the left ventricular ejection fraction. 50, everything above 50 is still normal. Between 40 and 50, that's considered slightly reduced, already pathogenic, and everything below 40 is a severe impairment. So how do these parameters correlate? What is the overall disease severity that we see? NT-proBNP, so the natriuretic peptide, is a very good indicator for disease severity, as indicated by the very strong negative correlation with the left ventricular ejection fraction. As you can see here, the inflammation measured as C-reactive protein is not that well correlating. A quality criterion for the management of the patient is that the more severe the disease, measured as peak anti-proBNP levels, and the stay in the hospital correlates also perfectly well. So our patients are very well taken care of. So what is the reflection of the inflammation if C-reactive protein is not such a good correlate? We performed a multiplex cytokine and chemokine assay called Luminex and measured other factors by Eliza and Christina did all these measurements. So what you can see here, this is a heat map with the healthy controls in gray and myocarditis patient from the two different cohorts in red. The heat map shows immediately for the many different factors that you can measure in serum that there is a tendency to not have inflammation if you're healthy, but also in some of the patients, and some of the patients show different varying degrees of inflammatory markers in the serum. So what is the dominant phenotype that we see? There are a number of factors, T cell activation associated factors such as interleukin-2 receptor, or other T cell attractants such as CXCL10 and CXCL9 that are consistently elevated in the serum in the impetiditis, so the Zurich and Gallen cohort, and in the control cohort from Tübingen. Other factors such as hepatocyte growth factor, which might be interesting for you if you haven't heard about that, or Adam. This is a factor that is produced by fibroblasts in the liver, but also the dominant source for hepatocyte growth factor are fibroblasts in the heart, and that shows a very strong relation and correlation with the disease severity. If we further delineate and segregate the different factors, the following pattern emerges. Namely, well, myocarditis is a T-cell driven disease, so all T-cell factors are significant. Almost all T-cell factors are significantly elevated in the myocarditis patient. Of course, some myocarditis patient with a mild disease course do not show such strong systemic inflammatory pattern. And some of the patients show both a macrophage activation signature and neutrophil CXCL8, IL-6, and the macrophage chemoattractant. But still, these patients in their serum show the presence of T-cell activation factors. So how to now mechanistically dissect this? It's a T-cell driven disease. Of course, there is a certain degree of analytical proceedings that you can follow in humans. So what you need is a mouse model or any in vivo or in vitro model that reflects the disease, the pathogenesis. And this is what we even had established before. Namely, we cloned a T cell receptor specific for the myosin heavy chain 6. These mice, we call them TCRM mice, develop a spontaneous autoimmune myocarditis that starts early on, right after weaning. Approximately 50% of these mice progress to what is now commonly defined as inflammatory cardiomyopathy, if not. And we have to euthanize them if the disease is very, very strong at the age of 8 to 12 weeks, but only a fraction, which is not that dissimilar as what we see in humans. Of course, the humans we all rescue because they are in the ICU as if the ICU is available, as I've indicated. So these are recent data, recent summary from Anna, also Anna, who is working under supervision of Christina. Of course, control mice, they live, and the TCRM mice, so the myocarditis mice, we have to euthanize them at different time points post-birth. There are two main disease causes, either mild or severe. Mild, low inflammation, low fibrosis. Severe, high degree of inflammation, high fibrosis. So the red staining is our collagen network. networks. In the mild disease, the heart looks almost normal, but there is always inflammation, and in the severe disease, there is dilatation and there is severe impairment. We also see this dichotomy in the phenotype in terms of fibrosis, if that is quantified. So the severe disease, it's this group, and the mild disease, it's this group, what you see here. So how is that reflected in terms of functional parameters? I've highlighted the left ventricular, the distribution of the left ventricular ejection fraction, so the heart function, the pumping of the function at admission at the first or second day in the hospital, and this is the spread of the ejection fraction in the TCRM animals, and the work has been done by Anna and Nadine. So this is an echocardiography of a normal, healthy control, and in the age-matched TCRM animals, what you can see there's an irregular beating. The ventricle, if you haven't seen these, that's what you see. This is the ventricle. The wall is thinned, and this is clearly a cardiomyopathy. That's a very busy slide. I will guide you very quickly through that. What you see, this is the time-resolved analysis of the ejection fraction, left ventricular ejection fraction in the TCRM and in the control mice. What happens early after weaning when all mice are still alive? So 24 of 24 mice are still alive. What happens is that there's swelling of the heart muscle, and that leads to changes in the function and with an significantly increase in the ejection fraction. So edema is known to affect ejection fraction, namely to increase also in humans. So after 6 weeks, I've indicated that some mice progress to the severe disease. So here it's 2 of the animals show a very low ejection fraction. We have to euthanize them. So hence we only have at that time point 22 mice left. Then we have an attrition in the cohort because more and more mice progress towards the disease, as you see here, and in the end there is a stable phase, what we call chronic myocarditis, where the values in the TCRM animals are not that dissimilar from the matched control animals. But what changes if we go further into detail? There is a measure, an echocardiographic measure for the cardiologist here that's simple. It's called strain, and that reflects the overall contractibility of the heart muscles of the heart muscle cells in different layers. I will not go too much into detail, but it can be longitudinal. So how does the heart compress in that way? How does the heart compress circumferential? And there's even a radial strain and the circumferential strain. What is the best predictor in these echocardiographic measures for the bad outcome? I spill the beans here. So it's the global circumferential strain, namely how does the overall ability of the heart to contract is a very good predictor at the age of 6 weeks for the progression to the cardiomyopathy, to the severe phenotype. I'll spare you some more of the data for the sake of time, also to have more time to discuss the conceptual summaries. So in acute myocarditis, once the first wave of immune cells get into the tissue, there's an increase in the ejection fraction, the edema, and there is the myocardial strain is altered already early on. The overall performance in terms of ejection fraction and strain normalize if there is adaptation to the inflammatory response. So if there is low inflammation and low fibrosis. So there's an acute phase, and then there's a normalization in terms of functional performance of function in the heart. If there's a progression to the inflammatory cardiomyopathy, ejection fraction drops because there's more inflammation and more fibrosis, as we've indicated, and the myocardial strain, in particular the the global and the circumferential strain change profoundly. So what is the mechanism and how does it work? And here comes Christina. And all that has been done before, and of course with Christian together. And that's the paper in 2019 that we had in Science, namely clarifying how does the changes in the heart that we have explained now, namely that there's loss of cardiomyocyte, replacement of the cardiomyocyte by fibroblasts. Right now you've seen it, including the functional consequences. How is that caused? And I've mentioned it before, we spilled the beans. It's the T cells working with the macrophages. So T cells have to get into the myocardium to exert their function, to lead to cardiomyocyte death, right? Highlighted here. And they have to get into the heart. They have to be activated, right? We go back in time or in the process to the root, and one of the root process in the activation of the heart-specific T cells is their activation in the heart-draining lymph node. This is where the myosin heavy chain 6 drains into from the heart, and dendritic cells present the myosin heavy chain 6, so that the predominant pattern of activation is what is called, immunologists call the Th1 phenotype, namely interferon gamma is one of the signature cytokines. So there's imprinting of a Th1 phenotype in the heart-draining lymph nodes, and a dominant pathogenic factor in these tissue-damaging reactions is the ability to secrete interleukin-17. Where does that happen? And that's the major conceptual progress that was delivered by this paper, namely that they're cross-reactive. Bacterial peptide mimic peptides in the commensal microbiome. Bacteroides theta iota omicron. There's a peptide. You will see the structure of the peptide in a second. That leads to then imprinting in the gut environment of the heart-specific T cells. And we know they're heart-specific because we cloned the T cells following immunization with the heart peptide. So it's the imprinting of a Th17 imprinting that happens via bacterial mimicry in the gut. So I promised the structures. So this is the murine and the human myosin heavy chain 6. They are functionally identical, and these are the identified, two of the identified mimic peptides. They're also very similar. This is the predicted binding to the IA of D. This is the class 2 molecule of the BALB/c mice, and this is, for example, the predicted binding of all these peptides to one of the more frequent HLA alleles in Europe, the DQ8. So overall, what do we conclude? It's the TCR repertoire, so the T cells that are present. Of course, the T cells need to be able to recognize their antigen. Microbiota have to be present to secure microbial mimicry, so antigenic mimicry. But of course, microbes are always good to steer up the immune system in general. And as said before, the genetic background has to fit, because if the peptide does not, this is immunology, if the peptide does not fit into the groove, then the T cells have nothing to regulate. So it's all consistent as well. Now, swinging back to the immune checkpoint inhibition, Javid Mosley and his team, they were among the first to describe immune checkpoint inhibitor myocarditis, and what they found in the few patients that they had analyzed was that the HLA-DQB10301 was one of the major, or they wrote, it's a New England journal, so everything's a bit focused. This is the major similarity between these two patients that they found, and this is one of the alleles of the alleles that is predicted to bind the myosin peptide and the mimic. Of course, that's all theory. How is that happening, and how does that all manifest in humans? So, due to we switch now back to the human, to the mechanisms. Is that really happening in the humans? So, for example, here this is published in Christian's Nature Cardiovascular Research paper. It's amazing that in these little snippets from the heart, we can detect 20, 30, 40% of the cells using single nucleus RNA sequencing being T cells. So that's hell of an inflammation in there. So if there is a high T cell infiltration, so a lot of activity, of course, a lot of T cells in comparison to other of these myocardial snippets with low T cell infiltration, the T cells are more activated, and of course, you would have guessed that whenever there are activated T cells, the best buddies, the macrophages, the inflammatory macrophages are activated as well. And in these regions, cardiomyocytes are really stressed, and there are a lot of molecules known to indicate cardiomyocyte stress and death are upregulated in these areas. Of course, these are high-dimensional data, and I just summarize it how we progress to analytically and hopefully therapeutically address this very interesting and we still think fascinating disease. So of course we're cutting it, right? I'm cutting many edges now, and I'm sorry for this complicated display, but I'll guide you through. So this is a multiple correlation analysis. What you would agree and what we've seen is that there's very high correlation, significant correlation between the influx of inflammatory macrophages and T cells. There are a number of factors that are then upregulated, downregulated, co-regulated. And here we picked and highlight the correlation between inflammatory macrophages and T cell infiltration with the downregulation of the bone morphogenic protein 4. Significantly, this downregulation is significant in these data sets. In the paper, you will see the summary soon of the Nature Cardiovascular Research paper. We had a few patient data available to confirm that this is as well seen in the serum of the patients. So healthy volunteers have roughly 100 picogram per milliliter BMP4, bone morphogenic protein 4 in the serum, and it's significantly decreased in the myocarditis patient. Of course, now, right, you have these findings, and you're always eager to see whether after 2 years it's still true, and if you double or triple the number of patients and it's still true, so BMP4 is significantly lower, so this is our extending cohort with the matched healthy patients, and the regulators, because bone morphogenic proteins are molecules from the TGF-beta superfamily. These factors in the tissues have to be regulated in a very tight fashion. And this is achieved by, of course, on the transcriptional level, but also by the local secretion of inhibitors. And the inhibitors of the bone morphogenic proteins 2 and 4 are called, funny enough, gremlins, and there are 2 of them, gremlin 1 and gremlin 2, and fitting to what we had predicted and what we had seen was that there is gremlin 1 and gremlin 2 significantly increase in the serum of the patient. And if we even now calculate gremlin 2 and BMP4 ratio, it's a very good predictive value here in the ROC curve as an area under the curve of almost 0.9, which is very good. And if you just have enough patients, then it's the p-values, of course, exponentially good. So we can also visualize that, and thanks, Christina, for visualizing that in such a nice way. So the gremlin 2, and we've mentioned that the natriuretic peptide, there's a very strong correlation between the gremlin 2 presence in the serum and this very good indicator for the disease severity in acute myocarditis. And the healthy individuals, as you can see here with the BMP4 in the bubble plot, are all low for BMP and gremlin 2. And for human data, that's a very nice correlation, and of course, highly significant. However, this is again, we're swinging now back. Correlation is not causation. It's good that there's strong correlations, but it's not causation. So what did we do, and what did Chris and Christina do in the lab? They generated an antibody against gremlin 1 and gremlin 2, and it's the antibody that we continue with is called 14D102. And how does that work? So I've mentioned it. So bone morphogenic proteins and the gremlins are important factors in the tissue. The receptors are called bone morphogenic protein receptors, BMP receptors, and if everything is running fine, and what is the function of many of these TGF-beta family cytokines is just signal the tissue everything's good, life is good, right? Have a tea, have a coffee, and just do your work and do what you're paid for. So that's homeostasis. Under inflammatory conditions, for example, in myocarditis, all kind of causes we will not go into, then the transcriptional activity of BMP4 and BMP2 is reduced, so there is less BMP4. Gremlins are even a bit elevated, so there is less BMP receptor signaling. That is, in immunology global terms, inflammation or danger that tells the tissue you are not in homeostatic conditions any longer. And at the locus of the fibroblast, that indicates and that fosters the activity of an inflammatory fibroblast. Well, now, if you intervene and you give an antibody, an inhibitor of the inhibitors, then you restore BMP4 availability, and with that, BMP4 receptor signaling. So I'll cut a long story short and show the key data by where Chris and Christina have treated TCRM mice, so the myocarditis mice with the antibody right at the onset, when the first wave of immune cells is attacking the heart, and you remember when there is edema, so acute myocarditis, and for 4 weeks, and after 4 weeks, the infiltration with immune cells is reduced in the heart, and what is important, the pathogenic cytokines, interferon gamma and IL-17, are reduced in the cardiopathogenic T cells. And what is important as well, the availability in the heart, so the heart tissues were minced and protein extracts were measured, and BMP4 concentration. There was only 3, right? Yes. I've got another 10 minutes, so that's very good. You know, I'm from Switzerland, although originally from Germany, I'm from Switzerland, and we are obsessed with time. If you've ever been to Switzerland, there are clocks everywhere. You just don't need to look. I don't have a watch. There are clocks everywhere. So whenever the clock rings, I get, am I too late? But and don't worry, ask Chris and Christina. If the bus is late, they really get nervous after only 15 years. I don't know how it was with Constantino, but you swung back, right? You're more relaxed now. So I'm still relaxed. So thank you very much for the nice intermission. So what happens now with the antibody? So we restore BMP4 availability, and with that, we'll not go too much into detail, fibroblast homeostasis. So we counteract the activity of an inflammatory fibroblast, which is conceptually still a bit new. People immunologists in particular, are of course familiar with the inflammatory macrophage, but that fibroblasts, the truly tissue-resident cells, can switch from one instant to the next into an inflammatory cell. This is what we are addressing here. So the next step, of course, is what do we do? And this is work of Hongwei Cheng, who's in the lab. If there is already a novel potential therapeutic principle, and you want to carry it into the clinics, and you have a mouse antibody, of course, you cannot give that to humans. The next step is to humanize the antibody. So we collaborate with a Swiss company called Lonza, and they have algorithms, techniques, and everything that is required for a process that is called CDR grafting, so the complementary determining region of those amino acid snippets from the antibody that determine the antigen binding have been transferred from a mouse antibody into a human antibody. That's the visualization of the antibody that has introduced a few more mutations. And what is always important to address and to assess whether a humanized antibody or a human antibody that has been extracted from the human repertoire is really functioning and doing what you want. Of course, it's helpful to have a mouse model that reflects and is very similar to the disease process that you would like to treat in humans. And the possibility that we have here is if we take mice that lack, so BALB/c, white mice, very similar in their genetics to the TCRM animals that lack T cells and B cells, we can transfer the cardiopathogenic T cells from the TCRM animals, so the cardiac myosin specific T cells. These T cells expand, they go into the heart, I've described that before, and they cause myocarditis. if we start the treatment with the antibody even before, and here it's a head-to-head comparison of the murine antibody and the humanized antibody, and wait for 4 weeks. What you can see here in the isotypes, so in the control antibody-treated animals, there's inflammation. In the treated with the murine antibodies, it's reduced. With the humanized antibodies as well. And what is important, that we get a significant reduction in the cardiopathogenic cytokines, namely interferon gamma and interleukin 17. So Christian and Christina, this is also, of course, right, the mission is to the reintroduction of you two guys into this microenvironment, into this ecosystem. So Christian and Christina have summarized that, and that was published last year in Nature Cardiovascular Research. And Pilar Martin from Madrid has written a very nice news and views on that. And to summarize that, overall, what I wanted to tell you, in the myocarditis and myocardial inflammation, it's the T cells, and it's the modification of the microenvironment, namely here at the locus of the fibroblast, that determines the extent of the disease, and at the same time offers opportunities for immunotherapeutical intervention. Namely that under homeostatic condition, BMP4, and probably other factors as well, produced by fibroblasts, signal into the environment, everything's good. Do your work, cardiomyocyte. Because why is that important in the heart? Of course, the work is done by the cardiomyocytes, but they are really hard workers. Many, many times per day is your heart beating and the cardiomyocytes are contracting. Plus, whatever you hear, cardiomyocytes do not regenerate. Once you lose them, you lost them. So the whole system has to be geared towards cardiomyocyte maintenance, survival, and well-being. That's simplified. So, and one of these well-being factors is bone morphogenic protein 4. Under conditions of inflammation, there's a reduction of that pathway. Fibroblasts are switching into inflammatory mode and foster inflammation whatsoever. And the antibody that the principle that we have discovered is to inhibit the inhibitor, namely an anti-gremlin 1 gram2 antibody, and that rescues, that antagonizes the disease and leads back to homeostasis. So I leave it here. I thank the whole team in St. Gallen. We have not that high mountains, and there are no volcanoes, at least in Switzerland, but the mountains are still very nice, as you can see in the background. It's one of your pictures, right, Christina?
Christian took it.
Christian took it, right? So thank you very much, muchas. So this is on one of the lab trips. This is the core group that is working with me. Christian, Christina, Hongwei I've mentioned, Nadine and Anna I've mentioned, Emily, Isabella as well. And we could not do the work with the team in Zurich with the support of the head of the clinic, Frank Rožička and Dörte. And there are other cardiologists and colleagues from all over the world who support our research, including the European Research Council and the Swiss National Science Foundation. And all that's left now is to thank you for your attention. Thank you.
Antes de darle la palabra a Christian, quiero comentarles que ellos están muy orgullosos, son egresados de nuestra unidad. Ellos estudiaron ahí desde la licenciatura hasta el doctorado y luego realizaron todos estos estudios, como lo mencioné. Y no solo ellos, sino más mexicanos, investigadores jóvenes, fueron a sus laboratorios a continuar su preparación. Entonces estamos muy, muy orgullosos y contentos y ojalá los podamos reclutar en un futuro próximo al instituto. Thanks, Burkhard, for this great talk. Now I'm going to handle to Christian to coordinate the questions.
De nada.
Yeah, there is one question.
Thank you for your presentation. It was fantastic. I have two questions. The first question is, what do you think that the T cells can change your phenotype in different severity levels in myocarditis disease? And the second is, do you think that inflammatory fibroblasts in this disease can drive this infiltrate of inflammatory T cells?
Yes.
Thank
so
you.
The answer is twice yes. I elaborate a bit more. So T cells are the drivers, right? Of course, there are different ways to affect T cell activity. However, our approach is more to go further to the root of the process in the tissue, because meddling with T cells has advantages and disadvantages. And we firmly believe that it's the future approach is to modify the microenvironment at the locus of the fibroblast. So the double yes, well, T cells drive, but the supportive infrastructure determines to what extent and how the T cells are acting, and of course together with the macrophage.
Answer, and the last question. Do you think that therapy over microenvironment like fibroblasts is necessary for this type of disease?
Well, that is, thank you very much. The answer again is yes. Interfering with T cell activity overall or with immune activity has been very successful, right? This is still what's given to these patients, prednisone initially, right? So you just hammer it, right? To come with the analog, with the war analog, right? That is, in terms of immunosuppression, the prednisone is something between between a very big bunker buster and a small nuclear bomb. So that's very severe. Still helps, but it's not a real long-term alternative, because inflammation in the heart in particular, right? Now we save these patients. The mortality was much higher. But the challenge is, if these, of course, identify the patients who are susceptible, in addition to the acute myocarditis, there's most likely, and there are very good indications, that inflammation in the heart muscle, is a major driver of more than acute myocarditis, of many other disease conditions. But right, as a very nerdy scientific approach, we prefer to study the processes in a patient cohort that is not at the end of a potentially immune-driven process, such as heart failure patients, cardiomyopathy patients, age 50, 60, 70 something, and with a very, very long list of medications already. So hundreds of confounding factors, almost impossible to delineate, to dissect pathogenic mechanisms. And hence, the focus on the acute myocarditis, where I've started, because these are young individuals, young patients who see the hospital first time in their life. And of course, cardiologists are asking, yeah, but we have to treat our heart failure patients, our cardiomyopathy patients. Yeah, of course. That's why the clinical studies in cardiology need 100,000 patients to see a minimal effect. And even in particular, the immune approaches many times have failed now. Retrospective analysis, sub-patient group analysis, oh yeah, but there are some patients that benefit. But the design has to change. Right? You can't run these clinical trials in cardiology in this way any longer. And it has to adapt to what is done now in rheumatology, or done in rheumatology for a long time, and what is done in oncology as well. Right? We have to treat and separate the patients according to disease mechanism, and of course to treat them early if there is a clear suspicion and clear indication that it's immune-driven. T-cell-driven, macrophage-driven, TNF-driven, or whatsoever. Sorry for the long answer, but that is from the bottom of my heart.
Hi, Burkhard. Thank you for that talk. I come from the Group A strep field research, which then goes on, repeated infections can end up in rheumatic heart disease. And very young patients, as you say. We don't have very clear, we know it's molecular mimicry. We haven't really identified the specific peptides that can be driving this progression of disease. But I'm curious about this IL-17 or Th17 imprinting. Do you know if there's something in particular in young individuals that can, have a more enhanced Th17 imprinting rather than adults? Because yes, as you said, it's like a young people.
No, we don't have data, but I would argue no, there's in principle no difference.
Yep. Right.
What is different in the more adult or more aged population is that the T cells overall get more experience, right? The thymus stops to work, there's no more output, and there's this clear selection of antigen experienced T cells. So the overall degree of what we now would call regulation is increased, so there is more regulatory activity in the more age-advanced population. So there are differences. But in principle, they still, I can still produce IL-17 or Th17 cells, because otherwise the bacteria would have eaten me alive.
Yep. Thank you.
Right, Adam? That's how it should be, right? Th17 should still be there, even at the advanced age, until the system breaks down. But there are questions already.
Thanks for your presentation. My question is, there a participation of autoantibodies in this pathology?
Sorry, there's?
Autoantibodies.
Autoantibodies. Oh yeah, autoantibodies are always present. The question is, to what extent do autoantibodies contribute to pathology? Right, we've heard that before. Antibodies are able to mediate via Fc receptors, complement activation, platelet aggregation. That's all possible mechanisms also affecting the heart. In the myocardial inflammatory disease in particular, antibody-driven effects are probably very rare. First of all, because B cells are very rare in the heart, and at least in the studies that have been done in mechanistically, autoantibodies or antibodies directly seem to affect the heart in a very rarely. So answer is yes, but probably not as severe as in other conditions. But still that has to be explored further.
And the second is, you are evaluate the Tregs?
Yes, we are on that, yes. We are confident that they follow the general rules with age increasing, and that disbalance.
The function is normal or affected in the context of the pathology?
That's it's overall right. We prefer not to look at Tregs alone, because it's the balance between the T cells that drive the disease and those that try to moderate and regulate the disease. So we always look at Tregs. So overall, in the T serum animals, the Tregs are not absent. On the contrary, 30+% of the cells in the inflamed heart and even in the severely diseased hearts express FOXP3 and qualify for regulation. But their activity is overruled by the Th1 and Th17 cells. And we would assume this is very similar in the patient.
Thank you.
Other questions?
So, Professor, you discussed 3 major factors that contribute to high risk for cardiomyopathy, which is like the TCR, microbiota, and genetics. Is there like a certain factor that we can work in for the future that can contribute the greatest to a decrease in the progression of the disease? And for the treatment,
please put your microphone a bit up. So
should I repeat, or?
I think I got it.
Okay. So also, should we use the anti-GREM for the treatment, or should a more generalized immunosuppression be used? Because if our goal is to save the cardiomyocytes, we can use the immunosuppression to get a stronger response, even though there is a higher risk of getting more infections. But I think cardiomyocytes, more cardiomyocytes can be saved that way.
Exactly. Yeah. So the way initially more broader, particularly in the very severe cases, the prednisone or maybe a bit more specific anti-inflammatory, global anti-inflammatory treatment is very important. Right? We have to cut it. This is right? Clinical experience has shown that works. But immune responses will continue, and there is a continuous, right? If you get it once, it's very likely that it's still cooking, right? It's smoldering. And we still have to find out, in particular in cardiac diseases, to what extent immune-driven smoldering inflammation is affecting the cardiomyocytes. And we see that it's a cut initially with immunosuppression, and then at the end find the most suitable therapy to maintain the environment most supportive for the cardiomyocyte. I think we would agree on that.
Great talk. And you show us the importance, the relevance on controlling this inflammation. I think this is great. But I want to go maybe a bit further on the resolution of the inflammation. Then I think there is something lacking there in the heart to resolve, and then it goes, instead to regenerate, it goes to the fibrosis. In our preclinical model of the axolotl, we find lots of, this axolotl, the Ambystoma mexicanum, it regenerates the heart. And then we find lots of B cells there. I mean, well, not exactly the B cells, but we find the VH and the genes at the moment. And I was thinking if there will be some role, and maybe we are lacking of protectins, resolvins, and all these factors that help, after controlling inflammation, resolve the inflammatory process. And
so resolution of inflammation is very important. We heard that before also from Adam. So control of the degree of inflammation in the heart in particular. It's even then different from or the equivalent to the loss of neurons in the CNS is, so how does the CNS cope with the loss of neurons? Because they cannot be replaced. So they grow more and additional connections, right? Axonal growth, additional connections. So how does that work? I would say compensation in the heart. The compensation is first that the fibrosis is not always bad. So the fibers that are generated by the fibroblasts that replace the cardiomyocyte are good because they secure the forced transduction, right? The cardiomyocytes have to contract, and if there are few of the guys are missing, someone has to bridge it, because otherwise there's no forced transmission. So that's what the cardiomyocytes do. Second, the fibroblasts and the fibrosis, in that sense, there has to be a remodeling in the heart so that maintaining the remaining cardiomyocytes that grow simply in size. What is the compensation? Right? In the CNS, it's more axons and more other connections, and in the heart, the remaining cardiomyocytes are just growing in size. So the infrastructure, right, coming back to what do we have to do? We have to optimize in long term the infrastructure in the heart to find out what keeps the cardiomyocyte happy, healthy, and protects the cardiomyocyte from further damage. That's also coming back to your question, and maybe answering partially what you just said. Overall, just starting from basic tissue biology and from the function. Good. Adam.
Hi, thanks. Really, really interesting talk. So my first question is the balance between the Th1 and 17 and in a systemic organ. And have you, I don't know, back-crossed the mouse onto like a Tibet or something like that, or raw gamma, and then done adoptive transfer studies just to see the relative contribution?
Yeah. We started with interferon gamma, which is it's a very interesting cytokine, right? It's pathogenic at the beginning, but if the interferon gamma or the interferon gamma receptor is lacking, at later stages of T cells develop as cohorts. They acquire on a cohort level different properties, and they adapt. So at later cohort maturation, differentiation, interferon gamma becomes regulatory and holds back So yes, we did. And you know, that's what you do in research, right? All the young guys here, the more you find out, the more questions you get. So yes, the cytokines change, regulation is kicking in order to keep, and it's very similar to what you observe, in order to keep the balance between protection, and here in the tissue, protect the cardiomyocyte at all costs, and at the same time get not minimize, but keep the inflammation at a reasonable level so that, again, the heart function is not affected.
And this is probably a naive question related in, I was actually quite surprised to see such a IL-17 contribution in a systemic site in terms of say to pathogens, to Salmonella, you'll virtually never see any meaningful systemic IL-17 or Th17 response. Mucosally, you will.
Yeah. So one explanation that we delivered for this question, surprise initially, right? IL-17 is important, is the cross-reactivity and the additional priming site, and it really happens that the heart-specific T cells proliferate in the gut-associated, in the colon-associated lymphoid tissues. Right? So there is priming, and once they're primed, they also leave the colonic patches, and then once they are instructed, they can go into the heart. So that is one, and we have other mechanisms elaborated or on the way, how the Th17 and where the Th17 imprinting in this particular context is following induction is also maintained and even amplified.
And just finally, a very provocative statement, you know.
I love that.
So MS, rheumatoid, but both of those are very, very strong T cell mediated component, but you treat with rituximab B cell depletion.
Mm-hmm.
So does that mean, you know, and probably that works, possibly targeting through antibody, but probably through regulatory B cells, which go not quite hand in hand, but you know, they know where each other live. And do you think that is the same, that the T cells in this not just are being pathological, but are being puppet masters? So if you target the T cells instructing B cells to do other stuff, you know, generic. Mm-hmm. So if you knock them out, you can maybe regain balance, or
So warning, it's a longer answer. Right? If you are, and this also relates to what we have with the regulatory T cells, right? If you assume that there is one dominant factor, and you call it regulation on the level of T cells, macrophages, B cells, whatever, and then you look for it, and then you find B cell regulation, and then you have your puppet master, right? But that's what we do. Right? We do the same. We postulate that there are inflammatory fibroblasts, and we find them, right? Because we had indication. The question is now, is that the only role? Most likely not, because B cells act on many different levels in the immune system. Without B cells, secondary lymphoid organs are not well organized, and you generate a relative immunodeficiency. Because the B cells are very important to maintain, for example, the splenic, the marginal zone in the spleen. Right? Without B cells, so you reduce the B cells, then the splenic structure is screwed up. Sorry, excuse me. And it's the same for the lymph node. So that's one. So it's the systemic immune reactivity. Second, B cells are very important antigen-presenting cells, and they're very important in the interaction with T helper cells. I would even say, well, they're at least as important as the famous dendritic cell. You may call them CDCs, PDCs, regulatory, whatever. Right? What is ever fashionable now, right? So the B cell therapy, and of course the experimental medicine that then shows that, yeah, well, if you cut, and coming back to the to global immune suppression, I think anti-B cell therapy is another means of very global immune suppression on many different levels. And it provides excellent opportunities for us as immunologists to postulate our regulatory cell population of the day, and to have a flourishing lab based on that. Sorry, that was sarcasm. Sarcasm off. But that's the way it is, right? That's we do it as well, right? We work on inflammatory fibroblasts and we focus on that, and we can because we need these conceptual narrowing, because we have to come up, right? Can we talk to the youngsters a bit, right? Because we have to come up with questions, we have to focus, and we have to ask clear questions. Because if we say everything is global, everything is right, then we don't achieve. So, in a very positive sense, yes, B cell regulation exists. B cell regulation is also affecting T cell activity. But there's more to it. Sorry, I declared the long answer.
Thank you.
Oh, do I get a prize? Thank you very much. Thank you. Thank you very much.
Thanks, Burkhard, for this great talk. Thank you, Adam. We really enjoyed your sessions and that you share with us this your part of your scientific journey and the latest developments of your research. I mean, I met these guys before the snow of time was flattering their temples, and it's great. They have been studying many different subjects and the evolution of the research and all the impact that has. I want to thank for the impact you have made here in Mexico with my students, with the students of other Mexicans, and all promoting research and help us to put forward immunology in Mexico, and in particular to take all these advancements to improve the healthcare of our people here. So thanks a lot, and thank you all for your presence here. Thank you for to the people that follow us from YouTube, Facebook, and the Academy website. It's great that you were here, and thanks a lot. Have a great weekend. Para los que están aquí, profesor Ludovic y el profesor Cunningham van a estar unos minutos por si quieren platicar, acercarse a hacer algunas preguntas. Vamos a estar unos minutos por aquí, después vamos a tener unas reuniones, entonces va a ser momento para que se puedan acercar. Gracias. So,